Hey students, this is Module 5, Cancer. Why do cells become cancerous? It's our first kind of lesson on the topic of cancer. These PowerPoints are in the modules in Canvas, so you can kind of open those up and follow along. Maybe take notes or something and look at them. These are the objectives for this chapter. These are the things that you should be able to explain at the end of the unit. This unit is module 5, 6, and 7 too, so the three modules here. So make sure you do all the guided practices and activities that are in those modules. Well, why do cells become cancerous? And then we'll talk about how does cancer affect my body. And then in module 7, we're going to talk about how cancer is treated. So, why do cells become cancerous? Well, first we need to figure out, what is cancer? Cancer is just a disease, a state of disease that's caused by the inability of the body to control the division of cells. Cancer can be triggered by certain viruses and certain environmental factors. It can be genetic too. But all it is it means that the cells will divide without really doing the job that they're supposed to be doing. There's some common forms of cancer like melanoma, leukemia, carcinoma, sarcoma, and then lymphoma. And cancer doesn't just affect elderly people. And it's the fourth leading cause of death in young people too. And males have a 1 in 2 chance of developing cancer during their lifetime. And for females, it's a 1 in 3 chance. Cancer is prevalent in humans. And so it's something that you kind of need to keep an eye on. So if you ever think that maybe you have something that could be cancerous, it's always a good idea to get it checked out. This is a pretty interesting chart here that shows male cancer death rates from the 30s up into 2015, and then female cancer death rates too. Why do you think lung cancer was really high and then come back down lower? And it's probably because of the rate of cigarette smoking in individuals there. So the body's organ systems have specific roles that maintain homeostasis, and homeostasis is this maintaining the same state within your body. And different body systems have different roles for that. Like the nervous system regulates the activities of all the systems. It responds to stimuli. The digestive system supplies blood with nutrients and water. Muscular system provides heat that maintains body temperature. And as the number of cancer cells grow, if, let's say you have cancer in the respiratory system, as those cancerous cells grow, it impacts the respiratory system's ability to maintain homeostasis or to help regulate the acid-base balance of the blood or to supply oxygen for tissues. And this is a quick check, look at these questions, kind of go back over it, see if you can answer them based on what we talked about and based on your reading. It's a good kind of study guide for your exams as well. So how does a cell normally work? Well, cells typically have the ability to acquire material and energy, respond to their environment by changing the internal environment in some way. Cells can reproduce through mitosis, and they can adapt to their environment too by changing things in the internal environment. I think we've mentioned before that cells can either be prokaryotic or eukaryotic. Prokaryotes are bacteria. This would be a eukaryotic cell here because it has lots of organelles inside of it, a nucleus, and a prokaryotic cell would not have a nucleus or any kind of these membrane organelles inside. So these are the eukaryotic cells that have membrane-bound organelles: nucleus, ribosomes, mitochondria, ER, lysosomes, all of these things that you can see kind of what their structure is and what they do. Cells are specialized too. During development, so as an early embryo or a zygote, you'll have an original cell. This cell will divide and you'll have multiples off of that one. These cells will divide and you'll have multiples off of that one. And then these cells will specialize for some type of specific purpose. Like this cell can become lung cells. This one can become pancreas cells. Actually, this cell can become anything. But if it becomes a pancreas cell, then these pancreas cells will always become pancreatic cells. And what cancerous cells do is they ignore their programming that makes them specialized, and they kind of lose their function. And so when you come back up in here and you look at, what these things are supposed to be doing, they just don't do it. And by losing their function, the body loses its ability to maintain homeostasis. Enzymes are usually protein catalysts for chemical reactions. The shape of the enzyme determines how it will interact with molecules. Enzymes have this active site where the substrate binds, and products are released after the reaction has occurred. And some cancer drugs work by binding to the active site, preventing the substrate from reacting with the enzyme, and therefore helping your body to kill off cancerous cells. DNA is a nucleic acid that stores genetic information. This is a molecule of DNA here. It consists of units called nucleotides, and this part here, so these kind of backbones, the ribbons that you see here, those are made up of this 5-carbon sugar and a phosphate group. All right, and this phosphate sugar backbone is the same over and over and over again. You can see there's the phosphate. So here's a little phosphate there, and here's a little sugar there. And they repeat over and over and over again. But inside here are the nitrogen-containing bases. And those nitrogen-containing bases are made up of, it's either adenine, thymine, cytosine, or guanine. And if you see an A, A's are always bonded to T's, and C's are always bonded to G's. Information is contained within DNA. This is what I just said here, and it's the order that these A's, T's, C's, and G's are in that stores genetic information of the cell. Right? The sum of all of this information is what we call a genome, and genes are units of genetic information that provide instructions for how to make proteins. And what happens in your DNA, you have this series of A's, T's, C's, and G's. And if you look here, this is a piece of DNA that's kind of straightened out. And one strand of your DNA that we call the template strand will have C's and A's and T's and G's. And what will happen is this piece of DNA here. So if you have, if you have a really long strand of DNA, let's say that we have a really long strand. I'm terrible at drawing this stuff. Sections of it, so maybe this part of your DNA is a gene that codes for, maybe it codes for insulin or something. Well, further down the line, maybe here, between here and here, is a different gene, and maybe this gene tells us how to build like thyroid stimulating hormone or something like that. And then further down on your DNA, maybe a really long section, is a gene for how to build, certain enzyme, like enzyme A or something. Well, these regions here, this one, and this one are called genes. And those genes are instructions for how to build this protein. And so what your body will do is it will look at this piece of DNA and it will make a copy of it, because you can't take the DNA out of the nucleus. So your body will come in here, it will look at this template strand, and it will make a copy. And the copy that it makes is called messenger RNA. And messenger RNA is not DNA, it's RNA. So it has A's, C's, G's, but instead of T, it's replaced by a U. Right? So C matches with G. And so you can see what we make is we make a copy, but we make what's called a complementary copy. It's kind of just the opposite of what's on the DNA. And then this copy will go out into the cytoplasm to these things called ribosomes. Right? And this ribosome will contain this messenger RNA, and the ribosome will help build a protein. Proteins are made of amino acids based on what this code is. And it will build this protein based on codons. All right, so this is one codon here. Maybe this is one codon here. This is the second codon here, and this is the third codon there. And because GGU, what you have is this transfer RNA carrying an amino acid. You need to remember that proteins are built with amino acids. Then it will form these long strands of amino acids, and it will string together into a protein. Well, this is what a protein would look like, and there are four kind of structures to proteins. The first one is this primary structure, or this linear sequence of amino acids. And a protein works because of its shape. Right? So the shape of a protein determines its function, and that's a very important thing to remember. So I'm going to type it up here. Protein function is determined by its shape. And because of its shape, well, that's terrible. So protein function is determined by its shape. Well, this order of amino acids that you put this in means that it will fold a very specific way. If I put this order of amino acids together by hand somehow and let it go, it will always fold, and it will always look just like this. Because a protein works because of its shape. Well, its shape is determined by the sequence of amino acids that you put it in. And the sequence of amino acids that we put it in is determined by the sequence of nucleotides on your genes. So therefore, if you change your genetic sequence here, you're going to change the order of amino acids. And if you change the order of amino acids, you're going to change how the protein functions. That's a very important topic. That's something that you need to remember. Here's another kind of quick check. Well, how do cells divide? So the cell cycle describes the organized series of events required for cell division. And this is a brand new cell here. This is a brand new cell here. This brand new cell is beginning its life. It will go through this G1 phase. And G1 phase is growth, occurs, organelles double. The cell kind of does its normal job. All right. And for some cells, it kind of gets stuck in G1, like nerve cells, muscle cells that maybe don't divide very often. But like a skin cell will go through G1, and after it spends some time in G1, it will make it to this S phase. Now, in S phase, the amount of chromosomes double. If this is a human cells have 46 chromosomes, and that's how many you will find in G1. But in after G1, in S phase, you will go from the beginning, you'll have 46, but at the end, what you'll have is 92. Because every chromosome in your body will then replicate. And then you'll go through this G2 phase where growth occurs a little bit more, and then you'll go through M phase where the cell will divide from one cell into two. Well, in this cell cycle, there are checkpoints where the cell kind of checks itself out. There's a G1 checkpoint, and if the cell's not ready for cell division, it will be put into G0 to attempt to repair DNA. And if it doesn't repair its DNA, then the cell may undergo apoptosis, which means that it will just stop living, basically. So we talked about your chromosomes. Chromosomes are these packs of DNA, compacted and organized. You have 46 of them. You have 23 pairs. You get one set comes from your mom, one set comes from your dad. And during the S phase, DNA replication occurs, and it'll look like this. Well, after the cell gets to the mitotic phase, several things will happen, and I'm going to kind of zoom this in so we can see it a little bit better. In interphase, we have some parts that we need to look at. In interphase is G1, S, and G2. So this whole part here, the orange, is interphase. You have centrioles, which are important, the nucleus, chromatin, which is your chromosomes, the nuclear envelope, and your plasma membrane. But once we enter prophase, which is right here in the M phase, the chromosomes are condensing, which means that they become tighter wound and they become more visible. Each consists of two sister chromatids. Right, so if this is a chromosome from your mom and this is a chromosome from your dad, let's make it a little bigger. This is a chromosome from your dad. Then whenever you copy it, you'll make one exactly like you got from your mom and one exactly like you got from your dad. And that makes these. So these are sister chromatids and these are sister chromatids, which means they're exactly the same. And what I mean by that is maybe there's a gene right here for eye color. And on your mom's chromosome, maybe she gave you the blue eye color. Well, there's going to be a gene for eye color here on your dad's too, but maybe he gave you a gene that gives you brown eye color, right? Well, all of these chromosomes are similar because it's eye color, eye color, eye color, eye color, but these are sisters because these are blue and blue, and these are sisters because these are brown and brown. And what we would call this is these are sisters. So this and this are sister chromatids. All right. But these four together are what we would call homologous. So these are homologous chromosomes. It means that they're similar but not exactly the same. So during prophase, we see the sister chromatids here. These centrosomes will begin to spread apart, and you'll get this spindle that appears in the middle. Prophase will continue. The nuclear envelope disappears. The mitotic spindle will then capture the sister chromatids at the centromeres that are holding them together. Oops. Then we'll enter into this thing called metaphase, where the chromosomes line up on the equator in a single file line. And during anaphase, they'll separate out. So these chromosomes go that way, these chromosomes go that way, and you'll end up with telophase, where where this will become one cell and this will become another cell. And the two new cells will have 46 chromosomes each. So why do cells stop dividing correctly? Well, mutations can happen. Mutations are changes to the nucleotide sequence of DNA. Here we see no mutation. CAC calls for this amino acid, GTG, this one, GAG, this one, so forth and so on. But in here, what's happened is you have an A that's replaced by G. Well, in this case, GTG calls for histine, but GTA also calls for histine. So we don't really have any changes in the protein. But let's say that we have here CAC instead of CTC. Well, if that happens, then you replace it with valine. And then here you have an abnormal protein. And sometimes you can replace a nucleotide that impacts the protein structure greatly by introducing a stop codon. A stop codon means that you stop building the protein, and in this case, this protein will not function at all. This one may still function some, but it'll function differently. This one usually stops functioning at all. Your body has tumor suppressing genes. They prevent unregulated cell growth. And let's talk about what unregulated cell growth is by looking at the cell cycle again. I'm going to erase this whole thing, so we'll do segment erasers. So here what we have is a kind of a cell cycle, and in this case, a cell will typically go through G1, spend some time in G1, spend some time in S, spend some time in G2, and depending on the cell, it could take days or weeks or maybe even months. And what a cancerous cell does is it will divide. It will almost skip G1 and go right to S, go right through S, G2 in just a matter of hours, or maybe just a couple of days, divide, skip again. And so this part here gets completely skipped. And this is your cell doing its normal functions. And that's not good. Because if you have a bunch of cells not doing their function and causing whatever system you have the cancerous cells in to stop functioning, then you can begin to have symptoms. Proto-oncogenes speed up the cell cycle. And so and onco refers to cancerous genes. And so these are genes that become oncogenes when mutations cause them to be stuck on. Sometimes a cell needs to be dividing quickly, but if a gene gets stuck on, then you have a cancerous cell. And there's a quick check at the end, and that is the end of Module 5. I encourage you to watch some videos on mitosis. I may post some for you. It'll explain, how mitosis works, and you can kind of visually see it without, my terrible drawings.